Stanford University.
Which allows to monitor the trafficking of cells. Dr. Contact is an associate professor of pediatrics and microbiology and immunology. He's an associate professor also in the pharmacology and is the director of the medical free electron laser program at Stanford University.
Thanks, Carl. So I was put in this position in the program today because we talk about, I'm going to talk about immune cell trafficking patterns as it relates to recognition of minimal residual disease, which is by definition comprised of cancer stem cells or tumor-initiating cells. In addition, this is a nice segue into the next session. Will be largely on immune cell trafficking and cell therapies. So we got into imaging because we had some questions that we couldn't answer by the conventional methods. We wanted to try and develop the tools that would allow us to watch what was going on inside the living body. I like this movie because it demonstrates some of the key principles of imaging, and I'll spend the first 5 or so slides talking about the tools of imaging using bioluminescence and optics. What I like about this movie is it shows you that you have now access to new information that you didn't have before. What we're looking at here is a transfection of a mouse liver with a reporter gene, asking how long does this gene express, to what level does it express, and so we're looking over a 6-day time course at 2-hour intervals and then 4-hour intervals, and demonstrating that we can monitor gene expression in living animals. So now we have access to new information because I'm looking at how a gene is expressed from a liver, using a liver-specific promoter in the context of the liver as opposed to hepatocytes in a dish. We get additional data for these studies because now we're looking at the entire animal. We're asking, did I miss my target? Is this gene expressed in the lung or someplace else? And so I can make some sense of the gene delivery tools. I can look at temporal changes, again, over a 6-day time course here. And if we have an imaging tool that really is easy to use, we can look with fairly fine time resolution. In this case, we're looking at hours, but you can also look at minutes, seconds, and now with some of the new instruments, milliseconds of how things change in living animals. In addition, now the statistics are better because I have built-in internal controls. I can tell you what this animal looked like 2 hours ago, 4 hours ago, 6 hours ago. I don't need to do separate groups of animals. And so I can look at change in individual groups of animals over time. I can use these images now to guide me to specific sites in the body at specific times to sample the tissue, so I can take the correct tissue at the right time, as opposed to sampling a black box, hoping I find the right tissue and sample at the right times. And we can use this now for image-guided genomics or image-guided omics because I know the context of how this gene or this cell is behaving in the living animal. And so we've done a number of studies looking and demonstrating that this gives us more information than we had previously. But I'm going to focus today on immune cell trafficking. But to do that, you need to understand optical imaging or the use of light in living tissues and what you really need to know to make sense of this data. You'll probably see all week long that people are using light to image biological processes without showing you the method or the essential elements of these methods. So what you've been seeing in that little movie in the previous slide is the use of this enzyme, which is derived from a firefly. It requires cofactors such as ATP and luciferin, as well as magnesium and oxygen. Luciferin is this molecule here, and if you inject this molecule into mice where all the cells in the body express luciferase, you can see that there are different patterns of distribution, whether you give an IP injection or IV injection. So when you think about getting this small molecule to this enzyme in every cell in the body, it's the same problem we have with drug delivery, but here you're actually watching real-time pharmacokinetics. And when we looked at these images initially, said this is great. Now we could use luciferin as a model small molecule. We could deliver it in nanoparticles, we could deliver it in liposomes, we could actually refine our drug delivery tools using reporter mice where everywhere that this small molecule or model drug goes, you get signals. Now you can develop new drug delivery tools. We spent about 10 years, as Carl pointed out, demonstrating that this small molecule crosses the blood-brain barrier, crosses the placental barrier, crosses cell membranes. But these movies are beautiful in that they demonstrate that it does indeed go everywhere. It also shows you what the kinetics of biodistribution are. And so if you take these little movies and turn them into just graphs, if you use an IV injection of the substrate, you deliver it to the tissues very rapidly, but you also clear very rapidly. If you deliver the small molecule with an IP injection, you have this plateau of about 5 to 20 minutes where the signal is fairly stable. And so these are important parameters to understand. If you're injecting your substrate IP but imaging out here, you're really looking at substrate distribution. You're not looking at the expression or the levels of enzyme expression in the cell target of interest. If you're giving an IV injection and you're imaging out here, you're also missing an opportunity. So understanding the biodistribution of the substrate and the cofactors is important for understanding what these images tell you. You may see some data throughout the week where we're looking at fairly large tumors that are likely hypoxic. Well, if this enzyme requires oxygen and ATP, you're probably going to get a diminution in signal as the tumor gets larger and larger. The other thing that's important to understand is how light goes through tissues. We just assume that since we put the reporter in there, we'll be able to see it, but it turns out that, of course, the light is attenuated as you go deeper and deeper in the body, which is why optics is probably not going to find its way into the clinic relatively soon, because we lose about a log of light for every centimeter of tissue, and if you're 10 centimeters in the body, you really have a weak signal. And I like this little experiment because it demonstrates that beautifully. This study was done by Brad Rice. We took a white light source, put it behind the mouse, and shined the light through the animal. And he asked, what color is the light on the other side of the animal? So it's a transillumination study where 16 different points on the animal are used, and white lights shine through the back of the animal, and then spectrophotometers used to read the color on the other side. I put the visible spectrum here just so you can see where we are in the electromagnetic spectrum. So what you'd notice is a couple of things. One, right at 600 nanometers, where the light begins to go from orange to red, you have this tremendous jump in transmission. This is a log scale, so it is indeed a significant jump. In addition, you'll notice that if you go through a fairly simple structure, like just muscle, the transmission is very good. If you go through a complex structure like here, where you probably have bone and intestine, the transmission is very poor. So this attenuation of signal is due to two things. It's due to the absorption by hemoglobin of light. Hemoglobin largely absorbs in the blue and green region, allowing the red light to transmit through tissues. It also is due to scatter, and that's demonstrated by the complexity of comparison of the complexity of these two sites, where the light is scattered as it hits different structures. So you're looking at this and you're saying, well, this seems a bit odd. You have a little bump right there. It turns out that if you plot the oxy and deoxyhemoglobin absorption, there's a little point here where you have a dip. So where the absorption drops, the transmission increases. What that suggests is that hemoglobin is indeed the primary absorber in the body, and I'll try and convince you of that in just a couple more slides. So here's that hemoglobin absorption spectrum for oxy and deoxyhemoglobin, again, plotted relative to the visible spectrum. And that little dip right here is where you saw that bump in the previous slide. And unfortunately for us, all of the reporters that we use that are bioluminescent have emission peaks right here in this region of the spectrum where hemoglobin has a tremendous effect on the transmission. So that you could look at that and say, well, that's good and it's bad. It's bad because the absorption is fairly significant. It's good because it's changing. And so you can ratio the amount of transmitted light at one point relative to the other and actually get some information about the depth of the signal. So one way you can do a 3D reconstruction using optics is to use the spectral information in the third dimension. And that allows you to do three-dimensional reconstructions. So as I said, all of those luciferases that we use emit in this region of the spectrum between 400 and 600. And these are representative enzymes that represent really all the colors of luciferases that we have available to use. This is Rhinella luciferase, green click beetle, firefly, and red click beetle. And some of you who read the literature may look at this and say, well, you're plotting your firefly incorrectly because in the literature it says that firefly has a 560 nanometer peak. Well, if you do it at room temperature, it does. If you do it at 37 degrees, there's a red shift. And so even with this firefly enzyme, there's about 60% of the light is greater than 600 nanometers. So here's that point where the transmission of light is actually pretty good, and both the red click beetle and the firefly enzyme emit quite a bit of light that are over the 600 nanometer cutoff point. And you can see that change in emission spectrum as you increase the temperature, going from room temperature to 37 degrees. Here's that 560 nanometer peak. But as you warm the enzyme up, it increases in its wavelength. It also increases slightly in its activity. The click beetle enzymes increase dramatically in their activity. So many times we'll do a study in an animal, measure the luciferase activity, take the cells or the tissue out, lyse them, and do the measurement in a cuvette, in a luminometer, and try and compare room temperature measurement here, room temperature measurement here to a 37-degree measurement here. And again, you have a difference because of just the activity of the enzyme is much greater at 37 degrees. And so by understanding how these enzymes function, you can better interpret your data and not simply work with the assumption that the enzyme is going to produce the same amount of light in each environment. And this is a beautiful experiment demonstrating exactly what I just described. We took 4 different enzymes, this blue enzyme, the green click beetle, the firefly enzyme, and the red click beetle, and we put them in 3 different sites in the body. We put them under the skin, we put them in the lung, and we put them in the liver. And then we scanned over the entire spectrum from 500 nanometers to 750 nanometers, asking, again, if you shine light from within an animal, what color is the light on the outside? And so we do the spectral measurements, much like Brad Rice did with his white light source in his animal. And if you don't have any tissue over the enzyme at all, you have really good transmission, even from this blue enzyme. As many of the blue enzymes are very bright. But as soon as you put this enzyme under the skin, the attenuation is dramatic. Now these arrows here are where we've normalized all of the curves. If I didn't do that, you wouldn't see any signal here at all. And I've plotted that on top of this hemoglobin absorption spectrum so you can get a rough idea of where the absorption of light is occurring and where the transmission of light is occurring. If you use a green enzyme, it sits right on top of this peak, these 2 peaks of the hemoglobin absorption. What I like about this curve is as you put this enzyme under the skin, the transmission perfectly correlates with the absorption, again showing that little bump between those 2 peaks, emphasizing that hemoglobin is the primary absorber in the body. And since we can't make hemoglobin knockout mice, we need to use enzymes that are red. And so the firefly and the red click beetle are about the same, and they attenuate both with absorption and scattering as you go deeper and deeper in the body, but there's a detectable signal that's possible. So with the bioluminescence readout, we typically use these red enzymes, and as you think about and hear about fluorescence imaging, you hear about near-infrared imaging because above 600 nanometers, really 600 to about 1100 nanometers, there's a window where our bodies aren't very absorbing and they don't begin to scatter, so we have that really narrow window of optical opportunities. So we detect these signals using low-light imaging systems. The first one back in the early 90s we built in my lab, and you can imagine a bunch of biologists throwing components together to make an imaging box. It worked okay, but it wasn't great. These instruments that I'm showing here are about 100 times more sensitive than the one we built back then, and they're much easier to use. The newer instruments have a lens turret, much like a microscope, so you can zoom in and out in the animal, and there's a filter wheel here, and that was essential for doing the spectral study that we did in the previous slides. And so these instruments have much more potential than the one we created in the early 90s. They do fluorescence and bioluminescence, and I'll show you only bioluminescence today. We do, however, link our bioluminescent reporters to fluorescent reporters so that we can do both flow cytometry and fluorescence microscopy with the fluorescence marker and whole body imaging with the bioluminescence marker. And we can do this by creating one protein with 2 functions using a flexible linker or internal ribosome entry sites, or the data I'll show you today is largely with a 2A ribosome slippage site where the ribosome will read the first open reading frame. You get pseudo-termination of the first polypeptide. The ribosome never disengages, but it reads the second open reading frame with the idea that you'd get equal molar expression of both markers. Now this could be reporter genes, or this could be a therapeutic gene in a reporter, but the idea is that the reporters would be equivalent in intensity. So in a study where we used a challenging model of lymphoma, and I say challenging because it doesn't grow in a dish, it only grows in animals. We can take the cells from the mouse, put them in a dish to transfect them, keep them there for a short period of time. And these studies were done by Matthias Edinger in collaboration with Rob Negrin. And you can select these green cells then and pass them several times through animals. And so when we did these studies with Rob, we said this is a great opportunity to really open up the possibility of looking at diseases that are cancer models that would be more difficult otherwise. And so to demonstrate that we had a good representation of the disease state, we did flow cytometry on these animals. We looked at 15 animals, either early in disease or late in disease, when you had metastatic lesions in the liver, the lung, and other places in the body. And we looked at the numbers of cells in the spleen and the liver. What I like about this study is that we had animals that we could see a signal in the spleen, but we couldn't really get enough statistical significance to plot those animals on the plot, so they actually plot down here. What this demonstrated to us is a very sensitive assay for in vivo measures of tumor burden. Not only was it sensitive, it had a very broad dynamic range. You can see the difference here. The acquisition times are 5 minutes or 30 seconds, so you can see the really dramatic dynamic range. If you count the cells in the liver and spleen, about 30 to 35% of the cells are these green lymphoma cells. In this animal, it's less than 1% in the liver and spleen. The data set from this blue animal plots here, the data set from this red animal plots here. There's 13 other animals in the study demonstrating that we have a pretty good relationship between the most exquisite cell counting assay and our in vivo measures. And so this seemed to be a very good measure of tumor burden based on flow cytometry. And again, the GFP allowed us to go back in and do the fluorescence microscopy at the end of the day. So you can put these lymphoma cells into mice, and you could treat them with whole body radiation, bone marrow transplant. You could show a reduction in the primary lesion, but you would see persistence of minimal residual disease. So we looked at this about 10 years ago now, so this is a tremendous, again, a tremendous opportunity to look at the cells that persist after conventional therapies. But we could also look in more detail at the cells that were in this bone marrow transplant, and you'll hear Rob Negrin talk about this afternoon, and you heard Irv talk about the stem cells this morning. And what I'd like to tell you about are some of the cells that we've worked on with Rob Negrin, the CIK cells that are also present in this population that are tumoricidal. And so we looked at this and we said, well, we have now a sensitive measure of tumor burden that persists after conventional therapy, and we don't need to isolate the cancer stem cell out. The mouse does it for us. If we treat the mouse, the cells that persist and give rise to the relapse are indeed, by definition, the cells, the cancer-initiating cells or the cancer stem cells. So we worked with Dean Felsher to create better models of minimal residual disease, and we've been working with Irv Weissman and Rob Negrin on this idea that if rather than whole bone marrow you put in hematopoietic stem cells, tumoricidal population, and memory T cells, you would have everything you need. You could reconstitute the animal, you could eliminate the residual disease, and you could at least have a transient immune response to the antigens that the donor had been exposed to. So you probably saw this yesterday with Dean Felsher, where we could create models using regulation of MYC, where we could inactivate MYC, create a state of minimal residual disease, remove the doxycycline from the drinking water, and get relapse. So we can look at each step in the disease cascade, from initiation of disease, late-stage disease, response to therapy, and then relapse. Imaging also gave us the ability now to look at cells that persist after fairly long periods of time and ask the question, what do those cells look like? What's the nature of minimal residual disease in this model? And I'm hoping Dean showed you this data yesterday, because I'm not going to go through it in any detail. But if you look at these regressed tumors, they begin to take on a normal liver morphology. This hepatocellular carcinoma begins to look more like liver. It forms the sinusoids of the liver, even though it's growing on the backs of these mice. The cancer markers are down, and as you see here, and the liver markers are restored. And if you did genomic hybridization on these cells, they have what appears to be a normal genome. So the cells that have this gross rearrangement of the genome don't survive in the absence of MYC. And as I showed you in the previous slide, these cells are metabolically silent. So everything that you'd use to recognize or kill a tumor cell, at least in this model, seems to be diminished or absent. So the question is, how do you eliminate a cancer cell that looks a lot more like normal? And I have to admit that we were probably a little bit lazy and didn't want to isolate all the cancer stem cells. So we said, why don't we let our immune systems recognize the cells and see if we can use that as a way to both identify and kill the cell, and also deliver additional oncolytic agents or tumoricidal agents. So we've been working on a population of immune cells with Rob Nager and the CIK cells that we know from our imaging studies would home to tumor targets. They would recognize abnormal vasculature, they would recognize a class of stress ligands, NKG2D ligands, and they wouldn't use a specific T-cell receptor. Because if we were relying on the recognition of a specific tumor antigen, that antigen may be downregulated in these states of minimal residual disease. So NKG2D is now emerging as one of an early marker of malignancy, and one that the immune system uses to recognize these early stages in disease. We know from Toby Schmidt's work, and Toby's sitting up front here, that it uses a unique set of chemokine receptors, and I'm not going to go through this data, I'll just summarize it at the end. And these cells are inherently cytotoxic. But the problem is, if you just give CIK cells alone, they're oftentimes insufficient to remove all of the tumor cells. And so we asked, could we add function back to those cells that would make them more lytic or cytotoxic? And so we thought we would add an oncolytic virus. The problem with oncolytic viruses is that they're essentially dumb particles that you inject. They don't know where to go. And so without any guidance, they oftentimes don't get to the tumor and are also insufficient. So we selected vaccinia as a potential oncolytic agent because it has a broad cellular tropism. We could study it in mouse models of cancer, we could study it in human xenografts, and it may actually have translatability. In fact, the vaccinia mutant that we've been looking at has been in clinical trials and showing really good efficacy with intratumoral injection. Again, because it's a dumb particle, you have to inject it into the tumor, it doesn't know how to get there otherwise. We reasoned, though, that if we could put this virus into these cells, that the cells would know where to go, and they would deliver, essentially do an intratumoral delivery rather than intratumoral injection of a very potent oncolytic agent. The genes that are deleted in this virus are thymidine kinase and viral growth factor, and that makes them tumor selective. And in any complicated combination therapy, knowing what's happening at each step is important, so we thought we could use image guidance at each point to assess the tumor burden, to assess where the immune cells were, and to understand how the immune cells were delivering the oncolytic agent. And what we discovered is that there's a synergy between these two therapeutics, and that we could image that synergy, and that's what I'm going to try and show you in the next few slides. If they advance, there we go. So we thought we could image all of these different components of the model, the oncolytic agent, the CIK cells, the infected CIK cells, the two mixed together, and the tumor, and we could do this with both luciferase, fluorescent dyes, and we could begin to pick apart each of the steps in this therapeutic intervention. The CIK cells can be enriched from both mouse and man in the presence of interferon gamma, IL-2, and anti-CD3. And if you inject those cells, those labeled cells, into tumor-bearing animals, you'll find that at some time after the injection of the cells, they seem to accumulate at the tumor site. And as I said, we're very interested in minimal residual disease, and so we use typically very small tumors, not large tumors, like I showed you in the previous couple of models. I was surprised when I saw this that it would take 24 to 72 hours for these cells to really accumulate at the tumor site. They indeed go to the spleen, they go to the lung and the lymph nodes, and eventually they're at the tumor target. So it's important to know how long it takes these cells to get there, because now we're going to try and combine a viral therapy with an immune cell therapy, and like I said, this virus is highly lytic. If indeed we put the virus inside these cells, it's likely to lyse those cells, and so understanding the kinetics of both viral replication and cell migration are important. The virus has these two genes deleted, so that rather than killing cells like a wild-type virus would, it spares the normal cells, but in cells where the RAS pathway or other oncogene pathways are deregulated and the cells are dividing and you have nucleotide pools, you can complement both of these defects. Thymidine kinase is required for viral synthesis, viral DNA synthesis, and the viral growth factor activates the cell to produce more virus. So we reasoned that creating this tumor-selective virus, we could selectively kill the tumor targets over the normal. And we could demonstrate this again with imaging. So we could take wild-type virus, and we got this from Gary Luker, or we could take double-deleted virus, this variant that I'll call now VVDD, or vaccinia virus with a double deletion. And we could inject those into mice, and the virus containing luciferase could be tracked. And if you look at ventral and dorsal images at 12 hours, they look very similar. So the two viruses look like they have similar distribution initially, lung, liver, and other places in the body. By 96 hours, the wild-type virus is replicating in those, continues to replicate in those sites, whereas the virus that has this double deletion has limited replication outside of the tumor. And so we could measure the tumor signal and show that the double deleted virus and the wild-type virus look very similar, and you could look at the wild-type in the other parts of the body besides the tumor and show that it replicates there, but this virus was indeed tumor selective. Not tumor exclusive, but tumor selective. And so the concept was we could enrich these cells. We reasoned that these cells would be rapidly dividing at this point, and if you put the virus in, the virus would lyse them. So we did a 2-hour incubation just to get the virus inside the cells. And then we would introduce this combination into the tumor-bearing animals and ask what was the effect on the tumor target. We've since learned that the virus grows very slowly in these cells, and I'll show you that data in a moment. It takes about 48 hours, so we could extend this period of time and actually improve the killing over what I'll show you today. So in a tumor target, the virus will replicate with very rapid kinetics. In 2 to 8 hours, the cells are lysed. In the CIK cells, even the wild-type virus grows slowly, but with the double-deleted virus, the virus essentially goes inside of the immune cell and sits there for 48 hours, not putting any viral proteins on the surface. And if you add either the virus alone or the virus plus the CIK cells, or the CIK cells alone to tumor targets in a dish, you see that each of these therapies kills the tumor target, but there appears to be a synergistic effect between the CIK cell virus combination relative to each of the individual therapies alone. If you drop virus directly on this monolayer of cells, you get in this case, a green fluorescence plaque assay where you're looking at the plaques forming from this virus. If you put the virus inside of the CIK cells and add those to the tumor target, you don't see virus initially, but with a synchronous release of virus, you infect all of the tumor targets all at the same time. So you have this explosive release of virus, killing all of the tumor cells, and there's nothing left at this 90-hour time point. And so part of the synergistic effect is the amplification in the CIK cell. We get 1,000-fold amplification and this uniform or synchronous release. And so we thought we'd try to demonstrate that first here in a 2-dimensional cell culture model, but then a 3-dimensional cell culture model where we're growing the cells, the tumor cells as a spheroid, and they're shown in red. The virus again has GFP, so you're seeing the virally infected tumor cells showing up here in green. And this is a plaque assay where you see the virus slowly spread through the tumor spheroid. And in this bottom movie, we've added the virus inside of the CIK cell. So you don't see any green virus because it's inside the blue CIK cells sitting on the surface of this tumor spheroid and not replicating to any great extent. There are some of the viral components that are being made, but it's not making GFP and it's not killing the CIK cells. So the CIK cells sit on the surface of the spheroid, and then they move in, and you see the 2 dye colors mix when they turn pink. And so there's still no virus being made, but right at this 48 to 54-hour time point, you'll see a synchronous release of virus. The entire tumor turns green and collapses. You see it right there. So the entire tumor turns green, and the tumor spheroid collapses. That's compared to the slow progressive plotting sort of spread of the virus here. So this is happening in a cell culture dish. If this were happening in an immune-competent animal or a human, the virus would be, the cell-free virus that's spreading through this culture would be neutralized by the neutralizing antibody. If any of you have been vaccinated for smallpox, you know you get this really weepy sore on your arm where you're vaccinated. That's happening inside this tumor, and there's a tremendous influx, inflammatory infiltrate if this were happening in an animal, and you make a neutralizing antibody that would destroy your therapeutic. In this case, there's I'll show you in a few moments, the virus is actually protected inside the immune cell. So we could go from a 2-dimensional cell culture to a 3-dimensional cell culture and then to an animal model. And we're comparing here the virus delivered by a CIK cells, the virus alone, in a rather large tumor. These are human ovarian cancers growing on the back of this animal. And by imaging, you notice a couple of different things. You say, well, the virus is great. Look at it, gets to the tumor immediately. And you don't see that with the CIK cells. They get diverted to the lung first, and then they go to the lymphoid tissues, and then they eventually get to the tumor, and you see that by 2 days. Still looks like the virus looks much better alone. Even at 3 days, it looks like the virus is much better than the CIK cell delivery. But you are seeing some off-target effects in this virus alone. If we use a green virus instead of a bioluminescent virus, we see that the uniformity of staining is better with CIK delivery than it is with virus alone. In addition, if you look at later time points, you see that the CIK-delivered virus seems to persist a little bit longer within the tumor. And so these are immune-deficient animals, so you're not going to see this neutralizing antibody overtake the virus. So we see a more uniform distribution, and we see what appears to be a longer-term replication. We can then use these images to guide us to where to sample the tissues, and we can do the histology, and this is a section through one of those tumors where you see the green virus at the surface of the tumor. You see the red CIK cells deep inside the tumor. The yellow CIK cells are those that are infected with the vaccinia virus, the green vaccinia virus. So the suggestion from this image is that the CIK cells are crossing the endothelial barrier and carrying the oncolytic agent right into the tumor. And this is further supported. If you stain for the vasculature, you see that if you give virus alone, the vasculature is what's turning green. So the virus gets into the endothelial cell and replicates, but never really gets to any great extent into the tumor. So you're not hitting your target if you give virus alone. If you use the cells to carry the virus across the vasculature into the tumor, you seem to infect many more of the tumor cells. And so synergy is in part due to this improved delivery, but we reasoned that there would also be molecular and immunological determinants of this synergy. And so we looked at these ovarian cancer cell lines. I think you saw the UCI101s in the mouse image a couple slides ago, but we also used SKOV3. I told you that the CIK cells recognize their tumor targets using NKG2D ligands. Well, SKOV3 is a very poor target for CIK cells because it doesn't overexpress these ligands. And you can see that here if you stain for MIC-A and MIC-B. The UCI101s are a great CIK cell target. They're killed very efficiently by the CIK cells, and they express the ligand. So we reasoned that since these are NKG2D ligands are stress markers, that if you infected those cells, you would upregulate NKG2D. And so there's no change in the cell that's already expressing a lot of these proteins, but in the cells that aren't expressing, you infect those cells, and now you're increasing the expression of NKG2D ligand, which is what the CIK cells use to kill the tumor target. So part of the synergy is also making the tumor a better target for CIK cell killing, and you can see that here. Here's the SKOV3s not infected. You infect them and they kill much better, and that's inhibited by an anti-NKG2D antibody. So that suggests the killing is improved, and it is indeed through that NKG2D ligand interaction. And so you can do these studies in animal models, and many times we don't show images of the animals. We show just all the data from each individual animal. This is the image data. This is animal survival. The dual biotherapy is a very effective therapy in these models. Each of these two therapies alone is okay compared to PBS. But this cell doesn't express NKG2D ligand, and so it's a very poor target for CIK cells. It's killed by the virus, and these animals survive a little bit better, and the dual biotherapy is improved. And you can look at each individual animal in these groups. Here's the untreated. Here are the dual biotherapy treated animals in the UCI101 animals, and in the SKOV3 animals, you see that there are some animals that are treated and some that are relapsing. The therapies by themselves, either the CIK cells here, or the virus by itself, is sometimes effective, but for the most part only controls the disease temporarily. So again, showing that there's a synergy in culture, there's a synergy in vivo. So now we're back to the question we wanted to answer in the very beginning. If you had a tumor model where you'd let the tumor grow, you would inactivate the oncogene or treat with a model, in this case a model drug, where you could reduce the tumor burden to undetectable levels. And then you would allow the animals to relapse. If you treated here, where the disease burden is low, and you can see that here with these images. And we also know that the molecular markers are more like normal because we've looked at those markers in the hepatocellular carcinoma and we've looked at them in the lymphoma model. Would you have the opportunity of eliminating the cells that persist at this point? Would the CIK dual biotherapy actually recognize those cells at this point, and even the cancer stem cells that exist at this time point? So here's that study. Lymphoma growing in the peritoneal cavity. This is the regulatable MIC model that we did with Dean Felsher. If you treat these animals with doxycycline, they all go into a state of remission. And in this case, the signal is so low we can't detect it. But we know that there are still cells that persist there because if you reactivate MIC, all of the animals relapse. And here you see the time course of that shown in days on top. If you give the CIK cells alone, you can control some of the relapse because, again, the tumor burden here is low, and the CIK cells manage to find some of the few remaining cells, and you can control relapse. If you treat with the dual biotherapy, you control relapse in most of the animals. There are a few animals that break through. And again, imaging allows you to look at the time course for each of these different groups, showing the peak of disease, response to the molecularly targeted therapy, and then the control of relapse in the dual biotherapy, where it's not in the individuals, there's less control. But even more importantly than looking at the groups, because we could do that with many other tools, we can look at each individual animal, like you would in a drug study, and show that without treatment, all the animals relapse. The CIK cells, you can control the relapse in some, but not all. And vaccinia is, like I said, kind of this dumb particle floating around. If it gets to the tumor, and this particular animal was lucky, it did get there in sufficient amounts that it killed the tumor. This one not quite so lucky, and the virus never got there in sufficient numbers. But the CIK cells can deliver the vaccinia virus to the tumor, and this is all done with a single treatment. We've proposed in subsequent, and actually have done subsequent studies in animals, that if you do multiple treatments, you can actually get all of the animals to be in this group and none of these early, middle, and late relapsers. So we're sitting here watching these animals, wondering if even after 90 days, if they would ever relapse. And we kind of got impatient, and we said, why don't we inject the tumor back in? 5 minutes? 3 minutes, okay. Why don't we inject the tumor back in and ask, would it grow at all? So we know from the cell trafficking studies that by 2 weeks, the CIK cells are gone. We know these are immune-competent animals, so in this case, the virus is all neutralized. There's no therapy left at 90 days. And so we just injected the tumor back in, and we could see the inoculum after injection, but all of the animals cleared the tumor. And so if we looked at the CD4 and CD8 cells, we saw that there's a very strong CTL response against the primary tumor. So in the process of lysing this tumor, in the presence of all this viral antigen, we've made a very strong CTL response against the original tumor. And this is one of my favorite experiments because I thought it was a failed experiment when we first started looking at the data, but it actually demonstrates this nicely. And we set this experiment up to try and model what we do in a clinical study. So these animals had very late-stage disease lymphoma. We treated them with the dual biotherapy, and you see that the vaccinia virus had an effect, the combined biotherapy had an effect, CIK cells had essentially no effect. And so we started watching these animals and saw all the animals relapse. We said, well, this has failed, it's not going to work. We said, well, Steve Thorne in the lab, who actually has done almost all of the vaccinia work in the lab, said, well, why don't we just treat here with doxycycline and turn the MYC oncogene off and ask what happens. And so he did that, and all the animals go into a state of remission, as you'd predict. But what was surprising in this study, and this is why I call it a rescue of a failed experiment, is that the PBS-treated animals all relapsed. And you see the error bars get bigger and bigger here as these animals die. But all of the animals treated with these biological therapies seem to have made a CTL response against the tumor, and many of them actually survive. And so this demonstrates again that part of the efficacy of this therapy is the fact that you're getting an immune response to the original tumor, and it's not simply the presence of the therapy. One of the big problems with oncolytic viruses is that you're worried about creating an immune response to the virus and eliminating your therapy. So we show here that inside the CIK cells, the virus is protected from neutralizing antibody. This is gamma globulin from the CDC. And if you pre-immunize animals with vaccinia, you still get an effective therapeutic response because the virus is hidden inside of the CIK cell. And this is further demonstrated in that study. Also, we've looked inside the tumor in a living mouse to show that the CIK cells are in the vasculature, probably the lymphatics, and into the tumor. And so we know that they're getting there, they're delivering the therapy. And even in animals where we've inactivated MYC and we have a state of remission, the CIK cells recognize where the tumor was. They recognize the abnormal vasculature. And I just have one set of summary slides left that kind of summarize everything I've told you and some of the things I haven't yet told you. But I'm not going to show you the data. I'm just going to do it in a cartoon form. The virus goes into the CIK cells, and it does replicate as viral DNA, but it doesn't make all of the viral components. So it kind of sits there in a quiescent state for 48 hours. The CIK cells recognize endothelial cells through CD18 and ICAM and some of the glycans that Jen Pressure in my lab is characterizing, and has a unique constellation of chemokine receptors that Toby has characterized that sense a chemokine gradient and are recruited into the tumor bed. The CIK cells recognize the tumor through this NKG2D ligand interaction. By this time now, the virus is replicated, and there's a 1,000-fold amplification of the virus inside the tumor. So you're delivering an amplified therapy right inside the tumor. That will infect the first set of tumor cells, and you get another 1,000-fold amplification. So you get intratumoral delivery and a 1,000,000-fold amplification of the therapy inside the tumor, and that then increases NKG2D ligands on the neighboring cells, increasing the NKT cell killing, and initiated a new CTL response that effectively eliminates that tumor. And so by combining in vivo imaging with cell culture assays, you can reveal the nuances of disease mechanisms and therapeutic response. And these visible animal models will actually probably have a greater impact on human health than will translating individual imaging modalities to the clinic, because many of these therapies can then be developed in mice and effectively translated to the clinic. Thank you.
For more, please visit us at stanford.edu.